Hello, my name is Tristan and I worked for Dr. Leah Goftis' lab this summer. Asthma is a common airway disorder characterized by inflammation. Airway epithelial cells are the lungs' first line of defense, where inhaled particles activate receptors on these cells, resulting in an inflammatory response. One of these receptors, Proteinase activated receptor 2 or PAR2, is what I worked on. PAR2 in the airways has been implicated in the development of atopy and asthma and has roles in initiating inflammatory airway responses. Previous work in the lab has shown PAR2 is upregulated in asthmatics and in stress cells. The relevance of this research is that improving knowledge on how endogenous asthmatic mediators affect PAR2 regulation can improve understanding of possible regulatory mechanisms in asthma. We hypothesized that pro-inflammatory cytokines will affect PAR2 expression in human airway epithelial cells. A virus-transformed airway epithelial cell line called Bs2b was cultured until 90% confluent. Some cells were stressed by removing growth factors for 24 hours, then activated for 24 hours using 3 cytokines with roles in asthma. Cells were lysed, RNA was extracted, and reverse transcribed, then quantitative PCR was performed. PCR data gave absolute PAR2 copy numbers, which were normalized using GABDH, then analyzed. IL-13, TNF, and IL-4 did not affect PAR2 mRNA expression in normally grown Bs2b cells. However, in Bs2b cells that had been stressed for 24 hours, as shown in the slide, we found IL-13 at 0.8 nanograms per milliliter and TNF and IL-4 at all concentrations significantly lowered PAR2 expression compared to base levels. IL-13 at 20 nanograms per milliliter also significantly decreased PAR2 expression when analyzing an increased sample size of 18. The significant p-values are shown as asterisks on the graph and explained below. Overall, our results showed that IL-13, TNF, and IL-4 at various concentrations significantly decreased PAR2 expression in stressed Bs2b cells but not normal cells. Knowing these cytokines are involved in asthma and that PAR2 activation can initiate the inflammatory asthmatic response, this is interesting. Possible mechanisms for why the cytokines affected stress cells is that there may be a different signaling or regulatory pathway involving PAR2 in stress cells compared to normal cells. Another possible mechanism is that stress cells have an altered sensitivity to these cytokines. Future work is needed to determine the reasons and mechanisms of these findings and to study other asthmatic mediators and their role in regulating PAR2. This may be an important mechanism related to asthma, which may give insight into future clinical work involving treatment of inflammatory diseases involving PAR2. Thank you.